So, first of all, a few notes. All the lectures, I send you emails. I want to remind you again, all the lectures and slides, please access via the NYU classes resources inside lectures and not through Zoom. I'm not, they are inaccessible through the Zoom recordings because I am downloading the lectures, I'm editing, I'm cleaning, I'm removing all the dead parts, I'm adding the timestamps and the links to the slides, and then it's easier for you to access everything, and it's more organized. I will try to do it faster, like tonight, to add already the edited lecture, not in the weekend. And I want at in the moment that I have more time, I want to compile one comprehensive, one document with all the lecture and the subtopics, with the links, with the time steps, so you can have one searchable document with all the information, and you can click and go directly to the lecture part. Updates. So, regarding our original timetable and all the mess with my absence. So I added one big lecture of psychophysics. It's 2 lectures condensed in one, edited. It's inside resources lectures. I hope, hopefully during this week, and I'm finishing another one about signal detection theory, which is usually not part of, in the last years, it's less and less part of the perceptual course. And in the book, it was sent to the appendix, but it's extremely important. It's a little bit more complicated, maybe. No, it's not complicated, but it has some technical stuff, but it's very important. So I want to add it there. Nowadays, when we hear about all the vaccines, correct rejection rates, false alarms, hits and misses, all these is part of signal detection theory. Visual attention, we began when we talked about reaction time and accuracy. We talked about the main paradigms, the flanker, the Stroop, the global local. So I will add, hopefully this weekend, if not later, again, 2 lectures condensed in one long edited lecture about visual attention. Today we are covering object recognition. On Wednesday we are going to cover color, and finally the grandmother cell stuff that I have been promising you. And then all we have is motion that I'll either squeeze during the next weeks, or I'll add as a complementary pre-recorded lecture, and we are back on track somehow. Okay, so today, after a short review, we will talk about adaptation, physiological and perception, and object recognition and its parts. So neurons, as we said, are very simplistic devices, and in different places along the path, each pathway of all sensory modalities, they converge differently to the subsequent afferent neurons, and that determines the sensitivity and resolution for different areas of the receptive area for each sense. So receptive field is the area in our skin or our retina or inside our eardrum. We'll cover that at a later part, or on our tongue. Each cell along any one of the pathways of all the sensory modalities, each cell has a very specific area geography on the receptive area of that sense. And we said that different, depending on the location, but it's not one by one, but on different locations along each pathway, and different modalities have more than one pathway, the receptive field of the neurons, they have different sizes. So in the V1, in the virtual cortex, in the occipital lobe, they can be very small, and the inferior temporal lobe, they can cover, in the case of vision, they can cover most of our, for one cell, cover most of our receptive, our visual field. Already very early in the pathways, cells start to have very complex and specialized configurations according to how they are stimulated inside their receptive field. So the on-off configuration is one that all our ganglion cells envision. They have different types of center on, center off cells. We also have the same configuration in touch. So specializations and spatial-based organizations, they are maintained, they are carried on along the processing pathways from the first initial area of receptors up to later cortical areas. Right. And we also agreed that neuronal activity is not perception. Information is interpretation of neuronal activity, and it's not the same. So, for instance, a simple example, but a very important one, is Bloch's law. Within a time window of 100 milliseconds, it doesn't matter how you present a given amount of photons, they are perceived the same. They are within 100 milliseconds, they are integrated, they are added to the same perception, okay? So if the area of all these, so this is the stimulus, all these areas are the same, and our perception is the same, even though neuronal activity was completely different along the pathway. And very important, we talked about lateral inhibition. That is a polarization of foreground and background. It's like increasing the contrast between foreground and background. It's a distortion of neuronal activity that leads to a completely different perception. It can be these distortions in a good way, it's not error. They can be spatially based, like in the case of our our ganglion cells in the retina. So the physical stimuli and the neuronal activity is different, is distorted, is different from our perception. All right, that's why we have all these kinds of illusions. And this polarization can also occur for higher level, more complex levels of perception that can involve much more meaningful interpretations of the words. So one example was the White's illusion. So while lateral inhibition, spatial-based lateral inhibition of the early stages of visual processing, let's say ganglion cells, we should expect to see these bars as lighter than these bars, but we see the opposite. Why? Because together with the spatial-based lateral inhibition, we also have a more meaningful perception of all this thing as a three-dimensional space. So we see these as one object hidden behind those bars, and these as one object on these black lines. And the lateral inhibition on this higher level of three-dimensional translation overcomes is stronger than the spatial-based lateral inhibition. That's why we see it darker, and it's a very strong illusion. We see it darker than this one. And also in color perception that we'll talk about, which is also non-spatial based. We'll talk about it on Wednesday. Okay, this is the last part of mechanisms of of the connection between neuronal activity and perceptual and perception. So adaptation. So first of all, we know, and if you didn't now, you will know that when a given neuron, one neuron, is stimulated in the same way during not long period for 1 minute, 2 minutes, if this one neuron is activated in the same way, the same stimulus falls on its receptive field in the same way, this neuron will gradually start to get tired or adapted, so its firing rate will decrease. And even if this stimulus stops, and after 30 seconds, 1 minute, depends on the neuron, depends on the sense, after a short period of time, the same stimulus falls in its receptive field, this neuron will react. And I'm not talking about chemical or chemical reuptake. It's independent of reuptake, the technical part. It's more complex. This stimulus will react less to the same stimulus. We know it from, even if we in our, I think that in touch we know it better. If you stimulate an area for in the same way, after a while it becomes a little bit numb. And if after 2 minutes again you stimulate in the same way, it's still a little bit numb. Okay, now let's see what does it make in terms of perception. So let's say this is one neuron that it specializes in these vertical bars when it falls within its receptive field. So here we can see when within these neurons' receptive field, when different orientations of bars falls, we can see the firing rate. It rises suddenly when its preferred kind of stimulus reaches its receptive field, okay? So this is an experiment to assess exactly the effect of this adaptation on perception. So a participant is given a stimulus on its receptive field without moving, and a dial. So the participant turns the dial that controls the contrast between the alternating bar until the minimum contrast that the bars can still be discerned. All right, so this will be the contrast threshold of the grating stimulus. So for this experiment, sorry, just a moment. We measure the contrast threshold for different orientations on that receptive, on the same area. And then we choose one, let's say the vertical one, and we present for 1 minute, 2 minutes, without interruption, which causes those neurons to adapt, to start firing less. And then we do the same thing with the same, we assess the contrast again for exactly the same orientations. And we see what happened to the threshold. And what we see is that for the stimulus that the participant was adapted, the threshold increases drastically. So when the participant turns the dial to the minimum contrast that he or she can still perceive the bars, look, it jumps drastically. Now, interestingly, we can see that it looks almost identical to the adaptation, the neuronal adaptation. One small difference is the tail. The tail of the neuronal adaptation is much more sharp, much more Gaussian, while the perceptual adaptation is a little bit wider. That's because the adaptation is not exactly, it's a little, so the, because of convergence and lateral inhibition, it ends up adapting also slightly similar orientations. How come when we look at something without moving our eyes, if perception if perceptual adaptation and neuronal adaptation are that similar, how come if we look straight ahead without moving our eyes, we don't start to stop seeing it? For many reasons. First of all, because we cannot really not move our eyes. We have all the time microsaccades. So saccades is when you move our eyes. We do it all the time. It's like a jerky move from fixation to fixation. But we have imperceptible microsaccades, like our fovea is like something that we cannot see with naked eyes if we look at the eye. Which in the foveal area, the same stimulus falls on slightly different areas within the fovea, because the fovea has a very condensed density of cones. With a high resolution, it actually falls on different ganglion cells. So we end up not having adaptation when we look straight ahead. But in the dark, if you try to look straight in very low light situations, when we use mostly our rods and peripheral vision, try it. If you look straight ahead at an object, after 30 seconds, 20 seconds, I told you about in the army that you look around. This is another reason. It completely disappears. What you are fixating in the dark, try it next time you are in the dark. Try to look straight ahead at one point. After 20, 30 seconds, it disappears. It becomes a blur. You have to look around to see it again. And also, not in a normal situation, in the same way that we cover, that the system patches up our blind spots, and for people that have astigmatism, which are distortions in the lenses, the system, by previous knowledge, by assumption, by inference, corrects the image or patches the blind. So the system does the same for uh, perceptual adaptation. Okay, object recognition. How can we perceive objects so well? We have actually, I'll cover it in a moment, we have very limited information in our that reaches our retina. How come we are so good in recognizing objects? So this is, in the last 10 years or so, a corpus of work by Molly Potter. Molly Potter, I'll say 30 seconds, I'll tell you about her. She's now 91 years old. She's a professor emeritus at MIT. She is my academic grandmother. My supervisor, my PhD supervisor, was her student at MIT. And she was born in Beirut. She's American. She was born in Beirut, and she spent the Second World War jumping all around the Middle East. She has a crazy, interesting story, and she's one of the most important names in the field of cognition and perception. She's extremely funny, and she's unbelievable. The last time I saw, I had many happy hours with this. Last time I saw her was in 2016. She was 86 years old, and she came and she gave talks and walks around. I haven't seen her since then. So this is a work that she many people started. I think she started with that. How fast, if you present a sequence of random pictures, but meaningful pictures, scenes that have a meaning, if you present one after the other, like how fast can we recognize the pictures? So she found out that if we present it as fast as 10 hertz is cycles per second, so 10 pictures in 1 second, like every 67 milliseconds, right? Or 133 milliseconds, right? Okay, very, very fast. It's like tick, 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 tick. We can 10 hertz. We can still perceive. We can still recognize those objects, and it's one after the other. It's somehow, if we have time, I'll talk about masking. We have a very a picture that comes after another picture masks the previous pictures for very strong periods of time, but somehow we are able to recognize these objects. Even though we have a couple of serious challenges. First of all, we have only two-dimensional pictures. All the information we have about everything are two very similar two-dimensional pictures that fall on our retina, right? The difference between the eyes is this distance, so it's nothing. We build one of the ways, we'll talk about it when we talk about that, is one of the ways we interpret three dimensions is the difference between the two eyes, but the difference is nothing. On the retina, a big object far away, presented in the same way, half of the size, half of the distance, retina-wise is identical, right? All the we can only interpret distances based on inferences and other clues that we'll talk about. The same problem is that two different objects, because it's two-dimensional, depending on the angle that they are presented, they can have exactly the same image on the retina. Some objects, most objects, depending on the angle that they are presented, they are technically, we cannot, they are unrecognizable. On the other hand, we don't perceive it. We do it very well. One example of this angle problem that we have is here. These are completely two different objects, right? Can anyone say what's the difference between this area, between this form and this one? Anyone in the chat? No, right? So look, they are identical. This is not a trick. They are literally identical. Okay. So our retina obviously cannot differentiate. How we feel, how we perceive it as two different tables, because of a lot of stuff that I'm going to talk about now. Another problem is that invariance is like we can see our retina can receive completely different images, and we automatically ascribe to the same perception. The perception is identical. If you are shown a series of images different, even three images one after the other, a duck, a chair, and a car, and then afterwards you are asked which of the viewpoints, which retina-wise are completely different, it will be difficult for you to recognize which one was the stimulus itself. You will remember the chairness and the kind of chairness, but not the stimulus. So it very fast is translated to something else. All these in a similar experiment, if you show a series of images, and one of them is one version of duck, you will remember duckness. Sometimes you cannot, depends on, well, this one probably yes, but okay, you understood the problem. When we have only a hint of the object, which is most of our perception in a day-to-day situation, we do it easily. Who can guess? Now play along, please. I know that you don't like it, but I implore you, use the chat or say it aloud. Who can recognize these people?
Well, we recognize Woody Allen, Bill Clinton, the dictator, Richard Nixon, and no, I'm not sure if that's...
This woman was married to this guy.
It's the Prince and Princess?
Yes.
Okay.
And this is Saddam Hussein.
Yeah.
Okay, so the amount of Charles and Diana, yes. So the amount of information that we have here is nothing. These are very small, and they are blurred, and still the gist is automatic. Like we immediately recognize. We don't have to search. We don't have to scan. So I suppose that many of you know it, but for those who don't know it, can you recognize it? Anyone? Anyone cannot see it? Please be generous with me. Play with me, please. Please. Okay. Can't see. Okay. Can you see a dog here? This is its head, the body, the legs. Can you see it? Yes, no, yes, vaguely. Okay, these, most people, the first time you see it, you still don't see. Oh, this is the dog. Can you see it now? There is a tree here. The dog is sniffing the ground. This is one leg, the black one. This is another leg. This is the body. This is a leg. Yes, yes. Okay. So if I wouldn't explain to you, probably most people would not be able to find the dog. But once you see it, you cannot unsee it. In 50, for the rest of your life, for those who saw it, you will never forget it. It's a crazy fact. This, you cannot uninterpret this almost abstract stimulus. So how do we do it? How can we organize things so well and so fast? So there are a lot of tactics, a lot of heuristics, a lot of concepts that have been proposed. So first of all, the Gestalt, you have it in the book. It's so simple that I will not waste our time talking about it. It's in chapter 5. Good continuation is something that from our knowledge of the word continues. Good figure is some coherence, similarity, proximity, common region. Even by the terminology, you already understand what it means. Helmholtz, this is from the early days of before psychophysics, when the first people started to try to formalize anything concerning before psychology, beginning of psychology. The idea of using Bayesian inference. So Bayesian inference is, I remind you, I hope that you remember, that Bayesian probability is the probability of a given event given the occurrence of another event. Okay, so what's the probability of when you throw a dice? What's the probability of a 6? It's 1 in 6, right? So what's the probability of having a 6 given that that it's the second throw and the previous one was 6? So Bayesian inference for perception is a formalization when the event is previous knowledge of the word, okay, past experience. For instance, what do you see here? It's an abstract nothing. But most people will see two rectangles with this green one. It's green, I don't know, I'm colorblind. This yellow one, it's blue, right? Purple one covering this green one. I'm kidding, I know it's red. So most people will see this. On the other hand, stimulus-wise, we should start with this. Okay. Another example was the White's illusion. We look at this and we immediately try to interpret in terms of a meaningful event, a meaningful scene. 3 dimensions. Okay, this is not in the book, and I think it's a very interesting theory. How much time do we have? Half an hour? No, 15 minutes? 15 minutes, right? Okay, so this is 30 minutes. Yes, good, good. So the recognition by components theory by Biederman, he suggested it in 87. I'll explain all of this soon. Can you recognize this? Yes. Anyone? Please. No, nobody can recognize this. This is a cat. So Biederman compiled what he called geons. He compiled 36 basic forms, geons, that the combination of them can somehow, not somehow, the combination of them can account, can construct the concept of most objects we know in the world around us, okay? So the attributes of these geons is like this. First of all, they have very distinct, each geon has a set of unique non-accidental properties, NAPs. So an accidental viewpoint is when the object, the geon, falls in a specific viewpoint that it cannot be recognized. So most viewpoints that you see a coin, you will recognize its essence, apart from the accidental viewpoint. So geons are highly discriminable one from the other because the non-accidental viewpoints of each one of them is unique. And these basic geons, they can be recognized, we can recognize them in most of the viewpoints. So they are different and they are very flexible in terms from which angle you look at them, you recognize its essence. They have very few accidental viewpoints. They have very few angles that you cannot recognize. So the principle of componential recovery is our ability to recognize an object if we recognize its componing, the basic geons that form that object. So for instance, yeah, even and that allows us, for instance, to recognize objects so easily when they are occluded, when they are partially hidden. For instance, can anyone, I will not give up, I'll continue to bug you until someone answers. Can you recognize this? What's behind here? No, you cannot. But can you recognize the same object? Yes, a flashlight. Yes. Now it's the same area. You have the same amount of information. The difference is the specific parts that allow us to recognize the geons that compose the object. So in a series of experiments, Biederman found out that objects like this airplane, objects that have 9 geons, we can recognize very well. 78% of the time we can recognize if we are given only 3 of the 9 genes, and 96% of the time if we are given only 6 of the genes. And objects with the 6 genes, with half of the genes, half of the information, we can recognize 92% of the time. Now, the recognition by component theory, it's a very interesting one. It can explain a lot of stuff, especially object invariance, the speed that we can recognize an object, a meaningful object, chunk it together with very little information, visual information, and the speed with it we can do it. But it's not a complete theory. It cannot explain other things. And it also, in terms of elegance, this theory, it feels that it matches what we know about the organization from basic construction of specialized stimuli, of basic forms that slowly construct a larger picture, like we saw in the visual system, in the ventral pathway. We see that first we have, for a very small receptive field, cells that recognize only this, but for this they do not, and then very different. And slowly the receptive field of the neurons, they become larger and larger. They have more shared area of the on the retina, and they start to react to a wider variety of stimuli. There are other theories. There is one very interesting one that actually a series of experiments that shows that actually when we are given a scene, we first of all, we recognize the gist, and only afterwards we start to scan and to collect the details of the scene. It's like go to the end and then go backwards. Okay, faces. Faces is a almost it's in the last 20 years, research on face perception is almost it is a subfield of itself. There are people that their entire careers, labs dedicated to that. PhD students, postdocs that work only with faces, only with specific effects of faces. Obviously, it feels to us that we have many reasons why, intuitively, why faces are so important in terms of Charles and Starr, in terms of evolution. So faces is one of the first things that that babies have to recognize, or they recognize as the first thing that they see all the time, and start to interpret what's going on depending on this blurred thing that comes in front of them. So one of the things that one of the strongest, so you heard about prosopagnosia. Prosopagnosia is the inability to discern between faces. Now, different people, we have a gradual ability, like different people have different abilities in recognizing, in discerning between faces. There are people that are extremely good. It's distributed gradually. So, and there are people that simply cannot. And until, I don't know, 20 years ago, I don't know, I'm just saying, until a certain point, nobody knew that there are, and there are people until today, they live their lives without knowing that they have this problem, because they use a lot of different information to discern between people. But if they are given, among you, there are people that if you are given a face without any other information around that face, you cannot recognize between two faces. Some people are better, some people are worse, but there are people that simply cannot. And we, even the people that are good in face recognition, by very small changes in the stimulus, we become prosopagnotic. For instance, if we invert the face. Now, what you see here, if you are given, if you are asked to discern between these three cars, you can be given two, one by the side of the other, and you can measure reaction time, or you can be given one probe, and then later you have to choose which one is the previous probe. So here we have cars, upright cars and inverted cars. We are more or less the same. For us, it's the same. We can recognize pretty the same efficiency. Faces, when they are upright, we are well, this difference is not because we are better. Here what we have is correct responses, okay? So the higher, the better we are discerning, we are recognizing. These, obviously, the difference between faces and cars depends on the stimuli itself. You can give very similar cars and it will be very difficult, or very different cars, it will be very easy, so it depends on the face. So we cannot compare between cars and faces. What we can compare here is the difference. If we invert cars, probably insignificant difference. This is probably statistically insignificant. But if we invert the face, look what happens. We simply cannot discern the face anymore. Which means, now, in terms of physical information, neuronal activity, we have the exact same information following on our retina. But apparently, the way we process this information is highly dependent on the way it's presented, for faces only. Can anyone recognize this guy? No? Yes, Denzel Washington. Can anyone see? This is a baby. Anyone see anything strange with this baby? No, right? Yes, a little bit weird. Here you can see that it's okay, it's the same people, but here we can already see, yes, the mouth and eyes are inverted. It's a little bit strange. I don't know. For me, Obama looks almost the same. Here I can see something strange. Now look at the baby. The inversion of the mouth and the eyes is crazy. When it's inverted, we can hardly see, because apparently our apparatus to recognize faces is so specific, so dependent on the presentation, then when it's inverted, we hardly, with the baby, for me, it's more strong. I cannot see anything strange. Here a little bit, but this is creepy. Not only that, these are newborns, 1 hour old. I don't know when they did it. 1-hour-old babies, they hardly can see anything. Many newborns, they hardly open their eyes. So they are shown schematic faces, not real faces, schematic faces, the same stimuli, okay? This is the control blank. This is the same stimuli, but not in a face form, right? You understand what I'm saying? So they are presented with this. The accepted paradigm for doing for experiments with baby is the time of fixation, for how long they fixate on a stimulus. You can assume whatever you want, but you see difference. So usually the main assumption is that the more comfortable they feel with the given stimulus, or the more interested, or the more whatever, the longer they will fixate on that stimulus. So look at the difference. In a normal face, they will, degrees of rotation, I assume, is their head. Yes, of the head and eyes. Okay, so it's fixation. For how long they remain fixated. So a newborn, it's hardly a human being. It already feels attracted to something that resembles a face. I find it very interesting. Okay, so not only we have a lot of these strong effects that seem to suggest that we have a very, very specific mechanism, machinery to recognize faces, which is very efficient, but very dedicated to faces, but very dependent on the way the stimulus is presented. And it was found that it also correlates to a very specific area. Now, this is controversial. That on Wednesday we'll talk more about it. What does it mean that we have a very specific area? So how did they find it? This was, it began, I don't know, 10 years ago. It was just before my PhD, I remember. So participants were presented with two images superimposed, one in one color, it's green and red, right? One in one color, the other in another. And they wore glasses with the same color filter. So they perceived, they saw the same stimulus, these stimulus, which are a face and a house superimposed one of the other, each one of different. But because of the filter of each eye, only one eye received each stimulus. So one eye saw only the house and the other only the face. Even though the perception, the person looking at the stimulus could not see, it was the same thing. And what it was found, and this is a technical thing, that how you can, with the inside the magnet, how can you find differences in activations of areas? So it was found that we have very specific areas, the FFA and the PPA, in one side, that they react, they are strongly activated only for houses, only for faces. There are some other objects, very common objects, but the strongest one is face. And the activation of the FFA is very strong when simulated by a face, and it's a very small. The voxels are very well determined. What we are going to talk, and I find it very, very, very interesting, very challenging, very annoying even, is the story with the grandmother cells. It began in 2005. I'll tell you on Wednesday about everything. I'm going to show you a 15 minutes movie, which was a talk that Itzhak Fried presented. Itzhak Fried is a now very famous, very important professor from UCLA and Tel Aviv University. Tel Aviv? Yes, I think Tel Aviv. And he and other colleagues, but I think he's the most famous name. He started with the story with the grandmother cells. And what he found is that they could find one cell. I'll explain on Wednesday how, and it's interesting how one cell reacts on one very specific concept. So the idea of the grandmother cell is that you have one cell that will get crazy every time your grandmother exists in your consciousness. It can be by visual stimulation, it can be by auditory stimulation, it can be by free recall. If you think about your mother, it can be, I don't know, I suppose, maybe smell, anything that reminds your grandmotherness, not to say different angles of pictures of your anything, and this cell will get crazy, and it doesn't react to anything other. Well, they made a lot of controls. We'll talk about it next time. And this is very annoying because, first of all, it touches on what I consider the biggest question, and I didn't have time with my long introduction, I wanted to arrive there. The biggest question that we have in humankind is the psychophysical question. I think that questions about life and death, they derive from the psychophysical question. The psychophysical question is, how can we connect the physical world to our self, to our inner experience? How can we connect any connection, anything between physical, between processing, electrical stuff, anything that connects between our identities, our feelings, and the physical world? And this is the most unanswered question we know. And in my opinion, everything derives from that question. And then Fried and his friends, they came with what? And memory. What is memory? Memory is everything in our experience. We can connect memory to everything we know. The opposite. There isn't anything in our experience that is not dependent on memory. Anything. If you think about it, without memory, there is nothing left in who you are, in how you feel, in how you think, in anything. And memory, we still don't know shit about it. I mean, we still don't know anything about it. There are many, the corpus of research about memory is probably the largest one in the field of cognition. And still, there is no one puzzle that connects. And then he comes with the grandmother cell, one memory. Now it's so annoying. We will talk about. Think about it. Think about it until Wednesday. And when you see, then I hope I will convince you guys to have a discussion.
I have a question.
Yes.
About object recognition when it comes to like line illusions. So like the shepherd elephant, the one where like you can't tell where the legs are, and things that are sort of like you can't totally identify it. I'm wondering just how those two interact.
Try to ask it in a different way. I'm not sure. I didn't understand the question itself.
Yeah. So I guess how there are illusions that are sort of open-ended. So like based on how you look at them, it looks like there are 3 prongs or 2 prongs because it's like the drawing is made to sort of trick you. And I'm wondering how object recognition comes into play with illusions like that. I might have to like pull up an example, but
If I understand what you're saying, well, illusions are exactly these uncontrolled interpretation. I think all of the illusions, they are a consequence of our need, the automatic system, the apparatus that cannot not infer meaning from a given stimulus. And either because we have not enough information, so the interpretation is wrong, illusion, or because we have more than one interpretation. And depending on how it's presented, our system falls automatically in one of the interpretations. Does it answer?
yeah, that makes sense. Thank you.
One, you know, the illusion about the vase illusion, when you have a vase and you have foreground or background. There was someone gave to Queen Elizabeth and what's his name, the Duke of Edinburgh. What's his name? Her husband. Anyways, so someone gave them a vase that the contour of the vase is her face and his face, each one from each side. It obviously only works in one specific angle. So all the contour illusions is an interesting one because you cannot see the vase and the faces at the same time. Our interpretation, our perception cannot cope with two simultaneous meanings, concepts, interpretations at the same time. We can move fast. Even if we train, we can move fast between the two. But we cannot perceive them both at the same time. There are all the illusions with the grandmother and the young woman, that the Freudian. Again, we can train and we can move from one to the other, but we cannot really perceive both at the same time, because the system it cannot not interpret. It cannot not give a meaning, even when there is not enough information, even if the interpretation is wrong, that's the system.
That makes sense. Thank you.